Hi again, and welcome back to another video on cellular respiration. In my last video, I covered the basic concepts of cellular respiration. If you haven't seen that one yet, I highly recommend you do so before watching this one, and I've included the link in the video description below. But essentially, we discussed how cells produce energy in the form of ATP from a single molecule of glucose, and how this process occurs through three main stages within the cell: glycolysis, TCA cycle, and electron transport chain. And in today's video, we'll be exploring the first of those stages in greater detail, and that is glycolysis. So what is glycolysis? Glycolysis is the very first step in cellular respiration, and it takes place in the cytoplasm of the cell. As you might have guessed from the name, glycolysis literally means splitting sugar. And that's exactly the core idea of this process. We're taking a single molecule of glucose, a 6-carbon sugar represented by the 6 blue circles in the diagram, and we're breaking that glucose down through a series of reactions. And as this happens, the cell is able to capture some of the energy stored in glucose in a couple of useful forms. So first, glycolysis produces a little bit of ATP, which the cell can use right away for energy. It also produces electron carrier molecules called NADH, which can store energy for the later stages of cellular respiration. And ultimately, the glucose molecule gets broken down into two 3-carbon molecules called pyruvate. And these pyruvate molecules then move on to the next stage of cellular respiration, where they can be further metabolized and processed to release even more energy. However, the actual process of glycolysis is a lot more complicated than what I've shown you here. And in fact, there are a total of 10 biochemical reaction steps in glycolysis, and each step is controlled by a specific enzyme that gradually converts one molecule of glucose into 2 molecules of pyruvate. And we'll be going through each of those steps in more detail later in today's lesson. But before we do so, it's helpful to think about glycolysis happening in 2 main phases. So we have the energy investment phase and the energy payoff phase. So what do I mean by that? Well, think of it like starting a business. In the beginning, you have to spend money up front in order to get it up and running. In glycolysis, the cell does something similar. It invests 2 ATP molecules in order to modify glucose and prepare it for breakdown. So that's the energy investment phase. And then comes the return on investment, the energy payoff phase. So now once your business is up and running, you can finally start making some money and generating a net profit. And it's the same idea happening in the cell. So in total, the cell produces 4 ATP molecules and 2 NADH molecules. So overall, glycolysis results in a net gain of 2 ATP molecules along with 2 molecules of NADH and pyruvate that can continue on to the next stages of cellular respiration. So before we get into it, how does glucose actually get into the cell? It enters through a process called facilitated diffusion. So that means glucose relies on these glucose transporter proteins on the plasma membrane, and it moves down its concentration gradient. All right, so now glucose is inside the cell. So the next question is, what does the cell actually need to do to glucose in order to make it usable for energy? So on its own, glucose is actually pretty stable, and that's good for transport in the blood, but it also means the cell needs to activate it before it can be broken down for energy. And this is where ATP comes in. So ATP, or adenosine triphosphate, contains 3 phosphate groups. So in the first step, what happens here is that ATP donates one of its phosphate groups to glucose. So this converts glucose into glucose 6-phosphate. And at the same time, ATP is converted into ADP. And this reaction is carried out by enzymes called kinases. So kinases transfer phosphate groups from high energy molecules like ATP onto target molecules, which in this case is glucose. And the kinase that is responsible for this specific reaction is called hexokinase. So hexokinases are expressed in most tissues in our body, but in some tissues like the liver and pancreas, there's another kinase called glucokinase that can essentially carry out the same reaction. But the key point here is that both kinases are capable of phosphorylating glucose. And once glucose becomes phosphorylated, it now carries a negative charge, which means it can no longer easily pass through the glucose transporters in the cell membrane. So as a result, it becomes effectively trapped inside the cell. And this is really important because it helps maintain that concentration gradient I mentioned earlier. So as glucose becomes phosphorylated, the level of free glucose inside the cell drops, and this creates a gradient that allows more glucose to move in from the blood. And in the second step, glucose 6-phosphate then gets converted into another molecule called fructose 6-phosphate. And this reaction is carried out by an enzyme called glucose 6-phosphate isomerase, which is also known as phosphoglucose isomerase. So what exactly do isomerases do? Well, they basically convert one molecule into another using exactly the same atoms. So in other words, we're not adding or removing any atoms. Instead, what we're doing is, or what this enzyme is really doing, is simply rearranging the structure of the molecule, changing its shape while keeping the same molecular formula. And this structural change is really important because it helps position the molecule to be processed more effectively in the next steps of glycolysis, where further modifications will eventually allow it to be broken down into smaller units. And another important thing to keep in mind here about this step is that glucose 6-phosphate sits at a major branching point in glucose metabolism. So while some of it can go through glycolysis for producing energy, some of it can also be stored as glycogen, which allows the muscle or liver cells to store glucose for future use. And some of it can also enter the pentose phosphate pathway, which is important for biosynthesis. But since this lesson is focusing on glycolysis, we'll continue on to the next step, which involves another round of phosphorylation. So much like the first step, the cell needs to invest a second ATP molecule, which donates another phosphate group to fructose 6-phosphate. And as a result, fructose 6-phosphate becomes fructose 1,6-bisphosphate, and ATP is converted into ADP. And this reaction is done by another kinase enzyme called phosphofructokinase 1, or PFK1 for short. And this is actually one of the most important regulatory enzymes in glycolysis because it basically helps control the overall rate of glycolysis depending on the energy needs of the cell. So when cells are low on energy, ATP levels drop and ADP levels rise. And some of that ADP can get converted into AMP, or adenosine monophosphate. So AMP serves as a strong indicator that the cell is running low on energy. So AMP can directly activate PFK1 by binding to an allosteric site on the enzyme. And this increases PFK's 1 activity, essentially hitting the gas pedal on glycolysis so that more ATP can be produced. Now, on the other hand, when the cell has plenty of energy, ATP levels are high, and in this case, ATP itself can bind to a separate regulatory site on PFK1 and inhibits its activity. And this may seem a bit counterintuitive at first, since ATP is also the substrate in this reaction step, but at high concentrations, it can also act as an inhibitor, basically slamming the brakes on glycolysis since the cell doesn't need to produce more energy. So overall, this is a pretty crucial rate-limiting step of glycolysis. And having said all that, what we're left with at the end is the formation of fructose 1,6-bisphosphate. So this molecule now has phosphate groups on both ends, which makes it more chemically unstable and ready for the next step. So in step 4, this molecule undergoes cleavage, and this reaction is done by an enzyme called aldolase. So essentially what we're doing here is taking this 6-carbon molecule and splitting it into two 3-carbon molecules. And these two products are glyceraldehyde 3-phosphate, or G3P for short, and dihydroxyacetone phosphate, or DHAP for short. However, there's a problem here. Only G3P can actually proceed through glycolysis, even though G3P and DHAP both have the same atoms and the same number of carbon atoms, which would make them isomers of one another. So the main difference lies here in how they are structurally arranged. So what do you think needs to happen with DHAP? That's right, so DHAP needs to be rearranged into G3P. And this reaction is carried out by another isomerase enzyme called triose phosphate isomerase. So what's happening here is that DHAP is being structurally rearranged into G3P. And the key takeaway from this step is that instead of having one usable and one unusable molecule, we now end up with two molecules of G3P that can both be used in the following step of glycolysis. So up until this point, we've been investing energy in the form of ATP through these enzymatic steps. And now we're going to shift into the payoff phase of glycolysis, where we finally start seeing the fruits of our labor and producing energy. In this step, there are actually two things happening at the same time. So now we have two molecules of G3P, and each one is acted on by an enzyme called glyceraldehyde 3-phosphate dehydrogenase, or GAPDH for short. And what this enzyme is basically doing is coupling in an oxidation reaction with a phosphorylation reaction in one step. So let's break this down in parts. So the first part is oxidation, where G3P becomes oxidized. And what do I mean by that? Well, G3P loses electrons, but those electrons have to go somewhere, so they're accepted by these electron carriers called NAD+. So in doing so, NAD+ receives those electrons and becomes reduced, forming NADH. And since there are two molecules of G3P, this produces a total of two NADH molecules. And this oxidation step is super important because it releases energy from G3P. And according to the law of thermodynamics, that energy does not get lost, but rather it's used to drive the second part of this reaction, which is phosphorylation. And so here, an inorganic phosphate group from the cytosol gets added to the oxidized G3P to form 1,3-bisphosphoglycerate. So now this is the first time we actually get to start making ATP during glycolysis. And at this point, we still have two molecules of 1,3-bisphosphoglycerate that we generated from the previous step. And this is a high energy intermediate molecule, so it can directly donate one of its phosphate groups to ADP to form ATP. And this reaction is carried out by an enzyme called phosphoglycerate kinase. So essentially, we took a phosphate group out of 1,3-bisphosphoglycerate and used it to phosphorylate ADP to produce ATP. And this is an example of substrate level phosphorylation because we're making ATP directly from a metabolic intermediate. And because we have two metabolic intermediates entering the step, we end up generating a total of two ATP molecules. So at this stage, we effectively break even from the two ATP molecules that we initially invested in the first three steps of glycolysis from earlier. And in the process, the remaining 3 carbon molecule gets converted into 3-phosphoglycerate. So now what we're doing is we're essentially rearranging the product that we got from the previous step in order to prepare it for the final energy producing steps of glycolysis. So in this step, the phosphate group gets shifted from the 3rd carbon to the 2nd carbon, and this reaction is carried out by an enzyme called phosphoglycerate mutase. So as a result, 3-phosphoglycerate gets converted into 2-phosphoglycerate. And now we move on to the second last step, and at this point, glycolysis is setting the stage for the final ATP producing reaction. So we start with the 2-phosphoglycerate we generated in the previous step, and an enzyme called enolase removes a water molecule from the substrate. And this type of reaction is called a dehydration reaction. So essentially what's happening here is a hydrogen and a hydroxyl group are being removed, and these come together to form water, which gets released. And that removal of water causes the molecule to rearrange in a higher energy intermediate form called phosphoenol pyruvate, or PEP for short. So now we finally reach the final step of glycolysis. And at this point, we have 2 molecules of PEP. As mentioned, PEP is a very high energy intermediate, so it's ready to donate its phosphate group, and that phosphate group gets transferred directly to ADP to form ATP. And this reaction is carried out by an enzyme called pyruvate kinase. And because we started out with 2 PEP molecules, this step produces a total of 2 ATP molecules. So overall, we've made a total of 4 ATP molecules in the payoff phase, But since we originally invested 2 ATP from earlier, our net gain comes out to be 2 ATP. And at the same time, now that PEP lost its phosphate group, the resulting final product of glycolysis we get is a 3-carbon molecule called pyruvate. And from here, pyruvate can enter the mitochondria to produce even more energy if oxygen is present, or it can undergo fermentation under anaerobic conditions. So to summarize the whole glycolysis process I talked about into a single slide, glucose is phosphorylated by hexokinase using ATP to form glucose 6-phosphate. Then glucose 6-phosphate isomerase converts glucose 6-phosphate into fructose 6-phosphate. Then another kinase, PFK1, comes in and uses a second ATP to convert fructose 6-phosphate into fructose 1,6-bisphosphate. And this gets split up into DHAP and G3P by aldolase. And triose phosphate isomerase converts DHAP into a second G3P molecule. And so each G3P then gets oxidized and phosphorylated by GAPDH to produce NADH and 1,3-bisphosphoglycerate. And next, phosphoglycerate kinase generates ATP as 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. And phosphoglycerate mutase then converts 3-phosphoglycerate into 2-phosphoglycerate. And enolase then removes water from the 2-phosphoglycerate to form PEP. And finally, pyruvate kinase converts PEP into pyruvate, producing another pair of ATP. So pyruvate can now enter the mitochondria to produce more energy under aerobic conditions, or pyruvate can undergo fermentation when oxygen is limited. And so overall, glycolysis gives us a net gain of 2 ATP, 2 NADH, and 2 pyruvate from a single molecule of glucose. Okay, so this wraps up today's lesson on glycolysis, and I hope this was helpful.